20. The industrialization of research and the war on cancer. The word cancer derives from the Greek carcinoma and the Latin cancer, for crab, suggested by the long distended veins coursing from lumps in the breast. During the first half of the 20th century, cancer was an almost unmentionable word. Public figures were never described as dying from cancer, and obituaries obliquely referred to a prolonged illness. An overpowering mood of powerlessness and fear, mixed with a sense of shame attached to the suffering caused by the disease, fueled a widespread cancer phobia. The ramifications of this pervasive fear are graphically described by James Patterson in his book The Dread Disease, A Cultural History of Cancer in the United States. Patterson includes an experience recounted by George Cryle Jr., a surgeon at the Cleveland Clinic. A 75-year-old woman who suddenly could not speak was referred to Dr. Cryle by her family physician, who suspected that thyroid cancer had spread to her brain. After a series of tests, Cryle informed the family that the disease was far worse than cancer, which he might have been able to treat. "There is nothing to be done," he told her adult children. "Your mother has suffered a stroke from a broken blood vessel. The brain is irreparably damaged. There is no operation or treatment that can help." After making this difficult speech, Cryle was taken aback by what came next. The oldest daughter leaned forward, tense and with a quaver in her voice, asked, "Did you find cancer?" "There was no cancer," Cryle replied. "Thank God," the family exclaimed. By mid-century, the stage was set for a divisive conflict over the nature of cancer research that would persist for decades. It involved a fundamental distinction between a targeted categorical disease approach and a basic research approach. The former requires a coordinated attack on a particular disease, whereas the second relies upon independent scientific studies of the processes of the human body. On one side of the conflict were those favoring centralized management and specifically targeted research. Even though such an approach typically involved a needle-in-the-haystack search for a useful agent, supporters were confident that triumphs were within reach. Others, particularly those in academic medicine, were highly skeptical of the role of regimented direction of biological research, fearing that it would lead to a certain loss of serendipity, and would favor a technological approach over independent, curiosity-driven basic research. As early as 1945, the Medical Advisory Committee, reporting to the federal government on a post-war program for scientific research, emphasized the frequently unexpected nature of discoveries. Discoveries in medicine have often come from the most remote and unexpected fields of science in the past, and it is probable that this will be equally true in the future. It is not unlikely that significant progress in the treatment of cardiovascular disease, kidney disease, cancer, and other refractory conditions will be made, perhaps unexpectedly, as the result of fundamental discoveries in fields unrelated to these diseases. Discovery cannot be achieved by directive. Further progress requires that the entire field of medicine and the underlying sciences of biochemistry, physiology, pharmacology, bacteriology, pathology, parasitology, etc., be developed impartially. Their statement, "Discovery cannot be achieved by directive," would prove to be sadly prophetic. At this time, there arose two figures who would play a commanding role in biomedical research in the United States: Albert Lasker, a wealthy advertising magnate, and his wife, Mary. As president of Lord & Thomas Company, the world's largest advertising firm, Albert had ironically pioneered for the Lucky Strike account the promotion of smoking by women in the 1920s and 1930s, with the slogan, "Reach for a Lucky instead of a Sweet." In 1942, the couple established the Albert and Mary Lasker Foundation to support biomedical research. It was based on organizing a small lobby of key professional and legislative people that would persuade Congress to allocate funds for a national effort. In 1946, the Lasker Foundation began giving out the highly prestigious Albert Lasker Awards in medical research. The most prominent spokesman in cancer was Dr. Sidney Farber, the scientific director of the Children's Cancer Research Foundation in Boston, and the discoverer in the late 1940s of the success of the anti-folic acids in combating acute childhood leukemia. Albert Lasker's death from colon cancer in 1952 reinforced his wife's determination that medical research provide answers, and Mary became an increasingly influential figure. She had energized a small, rather inert American Society for the Control of Cancer founded in 1913 by an elite group of surgeons and gynecologists into becoming the massive publicity and fundraising machinery of the American Cancer Society and added businessmen to its board. The U.S. government had undertaken support of cancer research in the late 1930s with the establishment of the National Cancer Institute, NCI. Even at this early stage, skepticism about the role of centralized management with regimented direction arose. Several doubted the wisdom of large-scale federal support for biomedical research. During the war years, it was a somnolent enterprise until funds from the defunct Office of Scientific Research and Development were transferred to the National Institutes of Health, NIH. Shortly after the war, Alfred P. Sloan, Jr., chairman of the board of General Motors, and Charles Kettering, GM's vice president and research director, gave $4 million to establish at Memorial Hospital in New York the Sloan Kettering Institute of Cancer Research. It became the largest private cancer research facility in the world. Cornelius Rhodes, its director, was known for his energy, initiative, and tenacity in the face of great challenges. He was driven by two compelling beliefs. Chemical agents could be found that would stop cancer cells from dividing, and research could be organized on the principles of the leading industrial labs of the day, like Bell Labs, which linked scientific inquiry with efficiency and high output. The industrialists who sat on his board encouraged him a great deal in the latter belief. Scientists at Memorial Sloan Kettering tested more than 1,500 forms of nitrogen mustard and other alkylating agents on animals and occasionally on patients between 1946 and 1950. Basic and clinical research was so industrialized that by 1955, about 20,000 chemical agents had been tested. Rhodes was featured in a Time cover story in 1949 as a confident investigator administrator and pictured with a crew cut and a white lab coat against a background illustration of a sword with serpents wrapped about its hilt, the symbol of the American Cancer Society, plunging through a crab, cancer. Rhodes zealously stated, "Some authorities think that we cannot solve the cancer problem until we have made a great basic unexpected discovery, perhaps in some unrelated field. I disagree. I think we know enough to go ahead now and make a frontal attack with all our forces." Some may have found his disdain for unexpected discovery, perhaps in some unrelated field, surprising in a man who had been involved with the serendipitous medical benefits of the military disaster at Bari just a few years earlier. By 1955, under lobbying pressures spearheaded by Rhodes and Sidney Farber, the center of drug testing had shifted from Sloan Kettering Institute to the NCI. The NCI was the jewel in the crown of the federally supported NIH in Bethesda, Maryland, which funds most biomedical research. The hopes raised by the enormous potential breakthrough that the two drugs, nitrogen mustard and methotrexate, could bring about cannot be exaggerated. It was believed that what sulfa drugs and penicillin had done for the scourge of infectious disease, chemotherapy, would do for cancer. In 1953, an article in Look magazine predicted that cancer would be conquered within a decade. Newsweek anticipated that a vaccine against cancer would shortly be developed. In 1958, Reader's Digest wrote approvingly of experiments being conducted on Ohio prisoners who had volunteered for injection of cancerous cells to test their immunity. The article considered this experiment a history-making research project that could lead to a breakthrough in the struggles to understand this dread killer. In the same year, Life quoted John Rod Heller, then the head of NCI, as saying, "I've spent many years in cancer research. Now I believe that I will see the end of it. We are on the verge of breakthroughs." By 1957, the chemotherapy program was immense, taking up nearly half of the NCI's budget and testing thousands of chemicals each year. Yet even the director of the NCI's drug development program, Kenneth Endicott, was highly skeptical of the approach. "I thought it was inopportune, that we didn't have the necessary information to engineer a program, that it was premature, and well, it just had no intellectual appeal to me whatever," he said a decade later. In 1971, the U.S. government finally launched an all-out war on cancer. In his State of the Union address in January 1971, President Richard Nixon declared, "The time has come in America when the same kind of concerted effort that split the atom and took man to the moon should be turned toward conquering this dread disease. Let us make a total national commitment to achieve this goal." As the country debated a bill known as the National Cancer Act, the air was filled with feverish excitement and heady optimism. Popular magazines again trumpeted the imminent conquest of cancer. However, some members of the Committee of the Institute of Medicine, a part of the National Academy of Sciences, which was asked by the NCI to review the cancer plan envisioned by the Act, expressed concern regarding the centralization of planning of research and that the lines of research could turn out to be the wrong leads. The plan fails, the reviewers said in their confidential report, because it leaves the impression that all shots can be called from a national headquarters, that all, or nearly all, of the really important ideas are already in hand, and that given the right kind of administration and organization, the hard problems can be solved. It fails to allow for the surprises which must surely lie ahead if we are really going to gain an understanding of cancer. The vigorous lobbying efforts, testimonies, and debates in congressional hearings in the 11 months before the Act's final passage remain highly instructive today and provide a template for the differing perspectives and dilemmas regarding the nature of creative discoveries. During the hearings, long-simmering opposing viewpoints were sharpened. A war within the war flared up over how much centralized control the proposed National Cancer Authority would exert. What is the best way to direct the path of meaningful research? Is it accomplished best under central direction, or should it be left more independently to curiosity-driven scientists? What role does serendipity play in medical discoveries? Can discoveries be anticipated and therefore targeted for funding and clinical trials, or do they often arise unexpectedly from basic research in a variety of disciplines? How important is the revelation of a fundamental finding in one field, chemistry, physics, pharmacology, physiology, to another? Is the term war in this context even appropriate, given that cancer is not a single entity and has multiple causes and manifestations? In numerous testimonies before congressional committees, Sidney Farber of Boston Children's Hospital had long served as a forceful spokesman for clinically targeted research, reflecting, it was believed, the immediate needs of patients. Impressive and dignified, he spoke deliberately and had a flair for the dramatic anecdote. Farber argued that it was not always necessary to know the cause of a disease in order to cure it or even to prevent it. His stand was vigorous. The history of medicine is replete with examples of cures obtained years, decades, and even centuries before the mechanism of action was understood for these cures, from vaccination to digitalis to aspirin. Sol Spiegelman, director of Columbia University's Institute of Cancer Research, stated that an all-out effort to cure cancer at this time would be like trying to land a man on the moon without knowing Newton's law of gravity. The emphasis, many declared, should clearly be on basic biological research rather than on centralized direction, which inhibits creative minds. A particularly compelling case was made by Francis Moore, MD, professor of surgery at Harvard Medical School and surgeon-in-chief at Peter Bent Brigham Hospital in Boston. Moore's opinion was diametrically opposed to Farber's. Moore's logical review of medical history at the hearings pointed out that if there had been a diabetes institute in the late 19th century, it would not have supported the work of Paul Langerhans on the pancreas, work that ultimately led to the discovery of insulin. At the time, the relation between the pancreas, insulin, and diabetes was simply not known. Similarly, he noted, a government institute on polio would likely have not supported the work of Dr. John Enders in the late 1940s in attempting to grow the mumps virus, work that found the method that ultimately proved critical to producing the polio vaccine. Moore argued that many medical advances come from creative research by, often young people, often unheard of people, in universities. Nevertheless, the Act was passed on December 23, 1971, signed into law by President Nixon, and hailed as a bountiful Christmas gift for the American people. When the dust settled, the National Cancer Institute was given substantial autonomy and markedly increased funding. The scope of the NCI's enterprise was breathtaking. By 1960, the program had begun screening 30,000 compounds annually for anti-tumor activity. These required almost 300,000 tests using 3 different types of mouse tumors. There were 2 major sources of chemicals: random, off-the-shelf selections and active programs of synthesis. By 1970, the potential of some 400,000 drugs had been explored. Over the years, the American Cancer Society tried to sustain the public's hope for continuous progress, often using phrases such as, "on the threshold of a golden age of medicine," and "a cure is just around the corner." At times, the evangelism even became reckless. In 1977, a former president of the American Cancer Society defended the society's aggressive promotion of X-ray mammography by declaring that, even though there might be a potential of increasing a woman's risk of breast cancer in the future, there's also an excellent chance that by that time, science will have learned to control the disease. In time, the odds against finding the needle in the haystack proved overwhelming. Only about 1 in 2,000 of those compounds screened for activity would be selected for clinical trials. The odds of a compound making it as a commercially available anti-cancer drug was at best 1 in 10,000. The NCI conducted extensive national clinical trials. Phase 1, generally involving a few dozen volunteers, determined the drug's toxicity and maximum tolerated dose. Phase 2, involving perhaps a few hundred patients, assessed any benefit and established the schedule of treatment. Phase 3 studies, typically involving thousands of patients, compared the treatment's safety and benefits with standard therapies. If the results were favorable, the drug would be submitted to the FDA for approval. Despite this Herculean effort and enormous expense, only a few drugs for the treatment of cancer were found through NCI's centrally directed, targeted program. These attack the fast-growing disseminated forms of cancer, such as leukemias and lymphomas, found primarily in young individuals. Over a 20-year period of screening, more than 114,000 plant extracts, representing about 15,000 species, not a single plant-based anti-cancer drug reached approved status. This failure stands in stark contrast to the discovery in the late 1950s of a major group of plant-derived cancer drugs, the vinca alkaloids, a discovery that came about by chance, not through directed research. By 1980, C. Gordon Zubrod, director of the Medical Cancer Program at the NIH Clinical Center, was forced to pay homage to the contribution of serendipity. He woefully concluded, "For the most part, the original discoverer of biological activity of cancer drugs was not aiming directly at discovering drugs for treatment of cancer. The original rationale for the biological study of a drug that later is proved to cure cancer may come from almost anywhere: basic research, targeted research, university, industry, but mostly from areas other than cancer. So one cannot program the initial discovery of drugs." Michael Shimkin, a longtime NCI physician investigator, in 1983 astutely likened the national cancer activities to the Strategic Air Command. 1/3 of the force is always in the air, coming and going to project site visits, reviews, conferences, and meetings in which the same people talk on the same subjects to the same audience. It is time to try longer investments in smaller laboratories and leave them alone to reach for the brass ring. In 1986, a report in the New England Journal of Medicine shattered the myth of progress and boldly declared that we are losing the war against cancer. This assessment was based on hard statistics. Combination chemotherapy had yielded amazing success against childhood leukemia, but the overall number of these cases was too small to have had any real impact on national cancer statistics. By this time, the government had spent upward of $8 billion through a vast national superstructure. One critic estimated that $1 trillion had been spent on cancer treatment and research since the beginning of Nixon's war on cancer. In 1997, biostatisticians John C. Baylor-Thur and Heather Gornick reported an updated and more sophisticated analysis entitled Cancer Undefeated in the same journal. This showed a 6% increase in age-adjusted mortality due to cancer from 1970 through 1994. The best strategy against cancer, they urged, was prevention, which could be promoted by convincing people to quit smoking and by urging doctors to use screening methods, such as mammography, pap smears, and colonoscopy, to detect cancers while they were still curable. Only Taxol, originally isolated from the bark of a yew found in the Pacific Northwest, finally achieved FDA approval in 1992 for use in the treatment of breast, lung, and ovarian cancers. Indeed, Taxol was the greatest treasure find in the government's 30-year screening program. Marketed by Bristol Myers, it proved to be the first billion-dollar blockbuster drug in the history of cancer chemotherapy. Overall, the few dramatic increases in cure rates and patient longevity occurred in a handful of less common malignancies, including Hodgkin's lymphoma, some leukemias, cancers of the testes and thyroid gland, and most childhood cancers. Sadly, these successes have not had a major impact upon the inexorable prevalence of cancer in our society, and the fact cannot be escaped that many of these successes came about in the early days of the war on cancer, or before war was even declared. The pap smear. In 1928, the Greek-American pathologist George Papanikolaou, while investigating the reproductive cycle by using vaginal smears obtained from human and animal subjects, observed the presence of cancer cells in people who were not known to be sick. That a cancer could be found this early through the examination of cells cytology was a totally revolutionary idea. Its elaboration is a classic example of medical serendipity by which an important discovery about cancer came from research in an unrelated area. Papanikolaou said the first observation of cancer cells in a smear of the uterine cervix was one of the most thrilling experiences of his entire scientific career. Papanikolaou went on to develop the famous pap smear test, so called from the first three letters of his name, which was not routinely adopted until the early 1940s. Cervical cancer, once the leading cancer killer among American women, is now, thanks to pap smears, a distant seventh. In June 2006, the FDA approved a new vaccine, Gardasil, made by Merck, which had proved highly effective at preventing cervical cancer in women. It works by providing immunity to two types of a sexually transmitted virus, human papillomaviruses, that cause 70% of the cases of the disease. By the mid-1990s, the National Cancer Institute redirected money to fundamental research into molecular processes, the relationship between diet and cancer, and prevention. The statistics, however, remain alarming. In 2004, almost 564,000 Americans died of cancer, and nearly 1.4 million new cases are expected to be diagnosed in 2006. In recent years, cancer has surpassed heart disease as the top killer of Americans younger than 85. In people 45 to 64 years old, cancer causes more deaths than the next three causes: heart disease, accidents, and stroke, put together. Such dismal statistics are all the more upsetting when compared with those for heart disease and stroke. Age-adjusted death rates for these diseases, which strike mostly older people, have been slashed in the United States by an extraordinary 59% and 69%, respectively, during the last 5 decades. Following the testing of nearly 500,000 drugs, the number of useful anti-cancer agents remains disappointingly small. Expressions of discontent with the methodology of research and the appalling paucity of results were, over the years, largely restricted to the professional literature. However, in 2001, they broke through to the popular media. In an impassioned article in the New Yorker magazine entitled "The 30 Years' War," Have We Been Fighting Cancer the Wrong Way?, Jerome Groopman, a respected clinical oncologist and cancer researcher at Harvard Medical School in Boston, fired a devastating broadside. The war on cancer, he wrote, turned out to be profoundly misconceived, both in its rhetoric and in its execution. The high expectations of the early 70s seem almost willfully naive. Regarding many of the 3-phased clinical trials, with their toxic effects, he marveled at how little scientific basis there was and how much sensationalism surrounded them. Groopman concluded that hope for progress resided in the uncertainty inherent in scientific discovery. Groopman's Pull No Punches article ripped off the curtain to expose the failings of those claiming to be wizards. The sad reality is that chemotherapy drugs are primitive, blunt-edged weapons of mass destruction. They interfere with the cellular process involved in the rapid divisions of cancer cells, but also act on other body cells with high metabolic rates and high cell turnover, including the bone marrow, intestinal wall, and hair follicles. Chemotherapy's ill effects on the bone marrow, the very foundation of the body's immune system, often result in blood deficiency diseases, diminished white blood cells, decreased platelets, aplastic anemia that lead to overwhelming infections. Furthermore, chemotherapy is of little or no use against many common cancers, including those that invade the gastrointestinal and respiratory tracts. It was never a very good answer to the problem of cancer. 3 years later, in 2004, Fortune magazine published a cover story lamenting the sad fact that we're losing the war on cancer due to a misguided approach. The emphasis, it asserted, should be on prevention and early detection. Interferon. Ironically, the NIH's effort in the 1960s and 1970s to find a cancer virus led to a discovery that was of great value years later in another field. By the 1980s, this effort had run its course. Only limited success had been achieved, most notably the discovery of virus particles in cells of lymphoma patients in Africa, the Epstein-Barr virus, the causative agent of Burkitt's lymphoma, and of an antiviral protein given off by cells when they were attacked by viruses. This protein, dubbed interferon, became an overhyped biotechnology product, but the viral work brought scientists a wealth of knowledge about retroviruses, which can copy their own RNA information into the DNA of the host cell. When HIV arrived in the United States, scientists were able to promptly identify the retrovirus, work out its biomechanisms of growth and replication, and target its cellular weak spots. The truth remains that over the course of the 20th century, the greatest gains in the battle against cancer came from independent research that was not under any sort of centralized direction and that did not have vast resources at its disposal. As we have seen, such research led to momentous chance discoveries in cancer chemotherapy and a greater understanding of the mechanisms of the disease that have resulted in exciting new therapeutic approaches.